Welcome everybody to the second installment of the Red Risk School, the Radiation Red Risk School. My name is Jeff Willey. I am Associate Professor of Radiation Oncology at Wake Forest and the Director of the Red Risk School. I'd like to thank you all for attending virtually during these times. A little bit about the Red Risk School. I see many of the attendees have attended before, but the purpose of this is to discuss the high priority risks to human health in space as determined by the Human Research Program's Integrated Path to Risk Reduction, designated by the color red. And radiation is certainly one of those risks. The degenerative effects of radiation on cognition, central nervous system, both alone and in combination with other hazards, many of which Dr. Stengel talked about yesterday. That's certainly a high priority risk for NASA. And so over the course of this week, we're going to be talking about various aspects of the risk. Now, as many of you know, our attendees come from a broad set of disciplines, from entrepreneurs to clinicians to researchers, academics. Many of you are interested in getting into the game and performing research related to improving human health in space. One of the platforms in which you can do that is through the Translational Research Institute for Space Health, so TRISH, who sponsors this program, who are looking for novel cutting-edge ways to address some of these problems. And so if you're interested in performing some of the research, what would be helpful is learning how to simulate spaceflight radiation. What resources are available for you to do that? Now, the past two Red Risk schools over the past 2 years, we've talked about the NASA Space Radiation Laboratory. But today what we're going to talk about is some other platforms that are available to perform spaceflight relevant research, radiation exposures, but also other types of exposures as well. These resources have many capabilities. And so we're going to be hearing from 3 content experts today. Now, before introductions, if you have any questions, please use the QA feature. We'll be monitoring those and we'll address that to the panel when we're done with all of the talks. So please use the QA feature. All of these talks will be recorded, by the way, and posted on the TRISH's website. So for today, we have 3 content experts. We're going to start with Dr. Guy Garty, who's an Associate Director of the Radiological Research Accelerator Facility at Columbia University. He's developed a wide range of radiation platforms, as well as leading development of automated systems for radiation biodosimetry. He's currently leading development of Columbia's preclinical heavy ion radiation platform, as well as a high-dose rate flash irradiator, which we, again, heard about yesterday. Next, we have Dr. David Smith, who is the Director of the Aerobiology Laboratory at NASA Ames Research Center. He's the Deputy Chief of the Space Biosciences Research Branch. Now, he has experience performing both ground-based, analog, and spaceflight-related spaceflight projects with payloads that he sent into orbit on shuttle, on ISS, as well as part of the NASA balloon program, studying in part how microorganisms can survive at the edge of the atmosphere. And then finally, we're going to hear from Dr. Mike Weil, who's a professor in the Department of Environmental and Radiological Health Sciences at Colorado State University. His expertise is in radiation carcinogenesis, and his research focuses on the individual susceptibility, and he serves as director of the carcinogenesis and score for NASA. So with that, we will hear from Dr. Garty.
Thank you, Jeff. So I'd like to tell you about the facilities available at the Radiological Research Accelerator Facility, basically simulating A-bombs all the way to doing microbeam research on single-cell targets. So who are we? RARF is a multidisciplinary accelerator facility designed for delivering known quantities of very precise radiation fields, primarily to biological targets, and we generally provide neutrons and ion beams. Of course, its energy is much lower than NSRL, but are very suitable for radiobiology. This is an accelerator facility that's pretty much dedicated to radiobiology, and we welcome outside users for doing experiments. So a little bit about our accelerator. We have a 5.5 million volt singletron shown here in one of our maintenance films. It's based around RF plasma ion source, which can generate beams of hydrogen, helium. We cannot fully strip heavier ions using this ion source. Then these ions are accelerated down a gradient of about 5.5 million volts, and we can use them to generate broad beams up to about 35 millimeters, or microbeams down to a micron or less for doing single cell irradiations. We're currently working on facilities for using heavier ions at higher energies, and I'm not really going to talk about our high dose rate electron facility, which is going to come online later this summer. In addition to our beams and our radiation fields, we have a fully equipped biology lab that's available for our outside users. We have a mouse facility that's a satellite for the main Columbia mouse facility, so you can do mouse experiments. We provide high-end imaging facilities, both on our microbeam end station and offline. We have a multiphoton microscope integrated into the microbeam. And we're in the process of building a SCAPE, or swept confocally aligned microscope for doing 3D in vivo imaging. And I'll talk a little bit about where that fits in later. And these fields are, radiation fields and facilities are generally used for 3 types of experiments. We're interested in neutron effects, in single cell radiobiology, and in ion radiotherapy. We also have projects involved in UV sterilization, which are very high priority at the moment, but I won't be talking about that. I'll start with neutron effects. And neutrons are not really identical to space radiation, but they're a really good surrogate for complex radiations encountered in deep space. And RARF was actually founded a little over 50 years ago to generate neutrons. It was founded by Drs. Rossi and Bond at Brookhaven because they needed a place to do microdosimetry of monoenergetic neutrons and to measure RBE values for neutrons. And Brookhaven donated the accelerator, which we were operating until 2006. And we basically, until 2006, and was replaced with the accelerator I showed in the video. We're currently located at Nevis Labs, which is about 20 or 30 miles north of New York City. In order to generate neutrons, we use an ion beam, typically either a proton beam or a deuteron beam hitting on a tritium target. And by adjusting the angle between the target and your sample, and the energy of the beam, you can adjust the energy of the neutrons. And that way you can get a monoenergetic beam for pretty much any energy you want between 0.2 and 14. And this facility is mostly used for physics research. We did some work on calibration of dark matter search, the Xenon 100 kilogram experiment. We've done measurements for the Mars Curiosity rover scintillator, and so on. For the biology experiments, we use a new facility that came online years ago, the Columbia IND neutron facility, which is aimed at modeling neutron exposures from an improvised nuclear device. And the model system we use is the Hiroshima bomb. And calculations have shown that within a survivable distance from such a device, you get about 30% of the dose due to MeV scale neutrons. So the aims we had in building this facility was to allow irradiation of mice and blood, and for developing of bioassays, and for testing radiation countermeasures. So basically you irradiate a mouse with neutrons, inject it with whatever drug you developed, and see if that helps survival. This again works in pretty much the same way. We have our samples on a fixture that rotates around our target at the center here. We use a beam of protons and deuterons impinging on beryllium, and then you get multiple nuclear reactions generating a broad spectrum of neutrons, which mimics the spectrum at Hiroshima at a survivable distance quite well. Dose rates, we can give up to about 3 gray per hour. We can, in principle, simulate microgravity for cell cultures by spinning them around very fast, randomizing the direction of gravity. Obviously, you cannot do that with mice. And we, of course, perform NIST traceable dosimetry for all our radiations. So that's it for the neutrons. A lot of the work we do focuses on single-cell radiobiology using our microbeam facility. A microbeam is a system for depositing radiation damage in a very localized manner on a micrometer region within a single cell. We've been using it to investigate single particle effects. For example, you hit with one single particle through a cell, see what happens. Looking at intracellular targets, and you hit the nucleus, you hit the mitochondria, and you see the effects that are generated. And also, probably the most interesting is looking at intracellular mechanisms of stress response, which are very important in low-dose exposures where you irradiate only a small fraction of the cells, but many of the cells that weren't irradiated also respond. How do we make the microbeam? You start with your biological target, in this case, a dish of cells. The accelerators are a radiation source. We have a particle detector downstream to count how many particles went through so we can close the beam when we've reached the prescribed dose. You need a focusing system. We use a 7 Tesla superconducting solenoid to focus down the beam to a 1 micron spot. You need an imaging system to see what you're hitting and where your targets are. And of course, a way to either move the targets to the beam or move the beam to the target. And you have to do everything under environmental controls because these experiments tend to take tens of minutes and you don't really want the cells to be unhappy being outside the incubator. And if you do this right, you get something like this. This is a cell line that has a GFP marker on one of the DNA response proteins, DNA damage response proteins, and we used alpha particle microbeam to write the name of our funding agency on the inside of a cell nucleus. Looking a little more scientific experiments, this is an experiment we did with irradiating mouse ears with a proton microbeam. In this case, we used a 35 micron diameter beam about this size, and we were able to show effects outside the beam up to about 1 millimeter. If you look further out, you don't get gamma H2X fluorescence, but in the vicinity of the irradiated area, even in the unirradiated cells, you do get activation of gamma H2X. So where we're going with these experiments is to look at ion radiotherapy. And to do that, we're interested in very high LET radions. And the reason they're very interesting is that they have a higher relative biological effectiveness compared to photons or electrons. And people have been seeing very encouraging results from Japan using carbon ion radiotherapy to treat locally advanced pancreatic cancer. And there are some indications that using high LET radiations, you get more induction of immune-mediated tumor killing. So this is something really interesting for the radiotherapy community. And the question that interests us is basically, is carbon really the best option? The mechanisms behind these immune-mediated tumor killings are not really well understood. And the question of whether you need carbon or a different type of ion can be a very expensive question. You can see here a picture of the Heidelberg carbon ion facility used for radiotherapy compared to a proton facility. Now, if you could get the same type of tumor killing with, say, a helium beam, you'd be much closer in terms of facility size and expenditure to the proton scenario than the carbon scenario. So we're interested in building a facility that will allow testing multiple ions, multiple ranges of LET on these sort of clinical samples. And this is also, of course, relevant for space radiation, both because we're generating end-of-track and secondary ions similar to what you would see in space, and also the LET range that we can provide to cell monolayers is close to what you have from galactic cosmic rays. So how do we do that? Our current ion source, as I said, it's an RF-based plasma source, can essentially generate proton beams, deuteron beams, and helium ions. Anything heavier than that, it doesn't have enough power to fully strip, and so we can't accelerate to high enough energies to penetrate into interesting samples. So to overcome this problem, we're in the process of installing a new Drebit heavy ion source in our accelerator. And the way this ion source works is you have a gas inlet that feeds gas into a magnetic bottle. There's an electron beam that goes through the gas, ionizing the ions, They then get trapped in the bottle and get further and further ionized until they're completely stripped, and then you can accelerate them out to the accelerator. And by having these fully stripped ions, you can accelerate them to the maximum energies possible for a given size of accelerator and get the maximum penetration. And we've shown on the bench that we can generate fully stripped carbon and nitrogen, and we're working on other ions between helium and carbon to find the conditions for operation conditions. So using that, we can generate a whole range of LET values. You can see here up to 1,000 kV per micron, but the problem is that the penetration is not very deep. So with the low LET, we can do irradiations of thicker samples or interesting samples, but with a very high LET, we can only do cell monolayers and not necessarily that. In order to be able to get more penetration, we need to increase our energy, and the way we're going to be doing that is to add a second accelerator in line with our singletron. So we've ordered a LINAC booster that will come here. You get your singletron giving 5.5 MeV or 2.5 MeV per AMU. The LINAC will double that up to 5.5 MeV per AMU, and then we'll get the sufficient penetration we need. And the way the booster works is actually quite cool. You have a series of rings with the AC voltage on them, and as the ion cluster goes between each two rings, it gets a little kick. And if you have enough of these in a row, you can get very high energy gains on your beam. This is a drawing of the ion LINAC that we've ordered and that is supposed to be arriving this summer. Most of it actually was a completed construction before COVID started, and we're still on track for getting delivery sort of midsummer. Once we get that beam up, we can increase significantly the penetration of our high LET radiations and allow experiments such as the one shown on the right, which is a radiation of a microtumor through a transdermal window in a mouse. To conclude, RARAF is a dedicated accelerator facility for radiobiology. We currently provide neutrons for cells, tissues, and mice, and hydrogen and helium beams for cell monolayers and thin tissues. Later this summer, we'll be able to provide heavier ions, and by the end of the year, we expect to be able to do heavy ion irradiations on thin tissues and possibly using windows in mice. High dose rates we currently can provide with protons right now, and we'll have an electron high dose rate facility about 200 gray per second later this summer. So typically we schedule our experiments on a month-to-month basis. You can contact me or fill out a beam time application on our webpage. And of course, I need to thank our funding agency. And most of our research and development is funded by various agencies of NIH. Thank you.
Thank you very much, Dr. Garty. While we get ready for Dr. Smith in his presentation, I'd like to ask one question which was posed on the Q&A feature, and we'll come back to many of these later at the end of this, but that is in regards to, you say that your facility performs a lot of performs studies with outside experimenters. Do you have many international collaborations currently, or has that been part of the portfolio in the past?
We've had international collaborations in the past, and we have them currently. I mean, we have people coming or sending samples from Europe. We have people coming from the U.S. Yeah, we're open to anyone who's interested in collaborating.
Perfect. And in terms of if a study is going to be proposed to work at your facility, how quick is the approval time, and just generally speaking, how busy is your facility? In other words, how quickly can it go from proposal to being scheduled to do the work at your facility?
We've in the past been pretty quick. If there's interesting science, we can get people on the schedule within a few weeks. Typically, if it needs to do either mice or something weird that requires approval through the university, it obviously takes a little bit longer. But for cell radiations, we've got a pretty fast turnaround.
Well, great. Well, thank you very much. And now we're going to switch gears and hear from Dr. David Smith.
Thanks, Jeff. Can you hear me and see my slides?
I certainly can.
All right, excellent. Thank you for the opportunity to present today. Thank you to TRISH. It's nice to see so many audience members. I apologize for the viewing format here. This is the only way to play my embedded videos, so hopefully the resolution's okay. Just bear with me on this one. I've got about 10 minutes of content. We'll have plenty of time for questions. Jeff mentioned, my name is David Smith. I'm a microbiologist and branch chief in NASA Ames Research Center and Space Biosciences Research Branch. Our branch historically has done a lot of space radiation studies, analyzing biological responses to both simulated space radiation and then actual experiments in low Earth orbit and increasingly deep space. It's really hard to get your experiments flown to space, as most of you probably appreciate by now. I'm pretty impatient and lazy, and I like to get instant results. I don't like to plan missions that take more than 5 or 6 years, and that's the reason I ended up starting to fly on NASA balloons. I want to teach you about how to do that. I want to kind of dismantle some of the preconceived notions you may have about balloons. The key word here is large in my title. These are massive scientific balloons that can lift huge payloads and linger in the upper atmosphere where near space radiation conditions can produce some pretty meaningful results for you to test experiments that you'd like to fly in deep space, or even to study biological responses for the environment you would expect, I'll argue, say on the surface of Mars when you look at the flavor of radiation in near space. So that's kind of where we're going. I will disclose I'm a biologist. Radiation is not my thing, and so I might have to defer to Guy and Mike and Jeff if you have any heavy physics questions. But I was interested in doing some biology experiments and kind of learned along the way of how interesting the ionizing radiation environment is in the Earth's upper atmosphere. So here we go. I mentioned I'm at NASA Ames Research Center. It's in Silicon Valley, California. Here's kind of the overview of what you've heard about probably before and what you'll hear about today. It's just comparing, you know, how we do space radiation studies, right? We can do them on the ground, like Guy was telling us. You can do them in space. Space Station, of course, is a great platform for that kind of research. It's just hard to access. And then up at the top, I'm showing you that beautiful view of Earth's atmosphere. And we can get balloons to get above about 99% of that atmosphere. And here's what it looks like when you do that. You can see the blackness of space out there. You can see how we've lifted ourselves up above. If you can imagine, this image is a very large scientific balloon. And this payload is attached to a big gondola that's kind of dangling below. And after the experiment, you come back down on a parachute. I'll show you some of that imagery along the way here. So this is where we're going. I want to characterize what we refer to as the near space radiation environment, paint the picture of how to do science on large balloons. I'll give you 3 examples of balloon payloads for any biological or dosimetry experiments that you may want to fly. And then I'll finish with a couple of examples of how to get on board, at least through NASA. I know we've got some international participants as well in the audience. So apologies to anybody I may be offending here, but I kind of want to just paint the differences between trying to do your radiation studies on the ground versus doing them in space or near space in the upper atmosphere. Depending on who is supporting your experiment, if you're paying out of pocket, say at NSRL, it's relatively expensive to get beam time. And if you can take a look at the bottom right panel, you're somewhat volume limited in what you can expose to the beam. And then I would also argue that the quality and dose you get is somewhat variable, and it's an imperfect simulation of the dynamic quality of radiation in space. And so consequently, the experiments tend to be relatively short-lived, small in order to fit within the beam. And a lot of the biological studies, if you look at the literature, are exposed in an acute fashion, which isn't representative necessarily of space. And so for all these reasons, I'm going to argue that we can use the upper atmosphere near space to produce a fuller, more natural, low dose rate ionizing radiation. And it also includes the secondary scattering effects, which are so important too for a lot of our studies. So this is a nice image from Mertens et al., which will help orient you to kind of what's happening in the upper atmosphere. Of course, we've got space radiation that's infalling, both galactic and solar. And in the upper atmosphere, so if you look underneath the magnetosphere there, where we're flying balloons, kind of above where that cartoon aircraft is, we have a pretty nice scattering effect of cosmic radiation that's falling and penetrating into the Earth's upper atmosphere. And we can linger there, as I've mentioned. So the balloon system is painted in this review article that Marianne So and I published a few years ago in Gravitational Space Research. And since I'm talking very quickly here, if you'd like to know more about the components of scientific balloons and kind of considerations for building payloads, please go ahead and take a look at that review article. It's freely available online. But you can see the various components on the upper right. And on bottom right, I'm going to show you just how stable the flight can be once you get into the upper stratosphere. So I'm going to play this video, and you can see one of our systems is activated in the middle portion of our flight. But that is an image of us floating in the middle stratosphere above New Mexico for about 8 hours. And you can see it's just a really nice, stable, continuously sunlit, radiation-exposed payload. And once the experiment has been terminated, in this case, after about 8 hours, we come back down softly on a parachute. And I'll go a little bit more into the particulars of this payload so you can see sort of how we load the biology and how we make both ultraviolet and dosimetry measurements on board this payload so that we can make some of the correlations between what's happening in this environment with the biology and the infalling radiation. So I'll keep moving quickly here. I've alluded to these balloons being massive. They truly are. So if you can see in the center of this image, Washington Monument for scale, when these balloons are filled at launch with up to 40 million cubic feet of helium, what happens is as they ascend into the upper atmosphere, they expand. And so on the right, you can see what the balloon system looks like at float. And then, as I mentioned before, dangling below is the balloon gondola with payloads, scientific payloads and instruments attached, and then a parachute as well connected for when the balloon is eventually terminated so that your science can come back down to the ground. A couple more overview charts from the NASA Balloon Program Office. Again, I don't represent the NASA Balloon Program Office. I'm a principal investigator that's flown with the NASA Balloon Program, and so I'm grateful to them for some of these overview figures. But what's depicted here is essentially, depending on how much helium you put in your balloon system, that determines how high you can fly and, in part, how long you linger. Also, how long you linger in the upper atmosphere depends on where you launch from. And so this is a depiction of the various launch sites from the NASA Balloon Program Office. Most new balloon payloads and instruments with the NASA Balloon Program Office will fly domestic missions over the United States first, either from Texas or New Mexico. Those tend to be relatively short-lived, 8 to 24 hours in duration, depending on what the meteorological conditions are like. However, if you want to get on long-duration balloon missions, you can launch from Antarctica or Sweden or New Zealand, and what happens there is you float with the jet stream. In the case of Antarctica, the polar jet stream basically does circles around the continent, and so you can launch very long-lasting missions up to 2 months. I'll say a little bit more about that later. First, I want to go back to the domestic missions. And again, this overview image from Mertens et al., who led an experiment at NASA Langley a few years back called RADEX. And the reason I want to bring up this mission for a couple of reasons, to orient you basically what the concept of operations look like for a balloon, but also they report some of the mid-latitude dosimetry, which is really important here for the case I'm trying to make, that the dosimetry of near space is pretty useful for people that are interested in, say, the surface of Mars. So I'm showing you there the average dose rates reported by Mertens et al. Compared to Hassler et al., who were making dosimetry measurements on the Curiosity rover. And they're awfully close, and I'll give you the punchline here. They're even better when you fly balloons from Antarctica. So here's another look from that mission RADX. This is from Norman et al. This is a series of papers that this team published in Space Weather in 2017. You can just see they're reporting here basically the different flavors of particles that they were measuring on that mission over New Mexico. So I'm going to keep moving quickly so we can get through everything here. I promised you I was going to show you what some payloads look like in case you're interested in either flying radiation instruments or biology. In my case, I'm a microbiologist. So what we do is we build autonomous boxes that are really easy to attach to the large balloon gondolas, such that we can load our biology inside these containers and then make very interesting environmental measurements along the way. We can power ourselves with an external battery as depicted in this image here for our latest payload called MarsBox, or we can plug into the power source from the balloon gondola itself. It really depends on the larger mission architecture. So I'm going to show you a couple more videos of our most recent test flight of this payload that was flown again over New Mexico in September. I'm going to show you basically how we activate our experiment on the bottom right. You're going to see the biology get exposed once we get into the lower stratosphere. You'll see the container swing open and expose our samples momentarily. There you go. Okay, and then once we get to the top most portion of the mission profile, somewhere around 36 kilometers, we lingered there for about 6 hours, and then on the way down, you're going to see our experiment terminate, essentially. So now we're coming down on a parachute, and you're going to see the little shutter there close such that our biology is protected for the descent, landing, and recovery. So that's just to help you understand kind of what a mission profile looks like. I'm going to give you a really sneak peek of some preliminary data. This is dosimetry results from our fantastic collaborators at the DLR in Germany. Thomas Berger et al. Has a fantastic active dosimeter called an M42, which was recently described in this 2019 paper you see listed at the bottom left. So again, you're seeing there some preliminary data of the kinds of particles that we're measuring, at least over the United States on a domestic mission. But I want to finish by pointing to Antarctic balloon flights, and the reason I want to do that is threefold. First of all, I mentioned that the flights can be longer lasting due to the Antarctic jet stream. Also, when you launch these missions in the Antarctic summer, you get continuous power, which you can see depicted in the middle part of this large Super Tiger payload, which we've flown with on 3 occasions. You can see the photovoltaic cells there so that you can continuously power your systems. So longer lasting flights, continuous power, but also continuous sunlight exposure, which is exciting for radiation experiments. And then finally, just due to the way that the magnetic field lines of Earth funnel space radiation particles to the poles, you're getting a really nice flavor of energetic particles. And again, this mission, the Super Tiger mission, was looking at cosmic rays, heavy cosmic rays, mostly iron particles they're interested in measuring that are infalling to Antarctica. And for somebody who's interested in space biology, I mean, that's just a perfect place to get smacked by some of those energetic particles. So again, you can see in the center of this image, we're just that little biology box. We're a mission of opportunity, or a piggyback payload that's stuck on board a much larger, more sophisticated cosmic radiation mission. And so here's what it looks like. This was our second flight from Antarctica. In late 2018, you can see the Antarctic ice well below us there. You can see our box that we call EMIS, that payload in the central portion, and our biology that gets exposed on command, and some of the radiation sensors in the central portion of the payload. Again, this is what the payload is. It's called EMIS. This is just a quick overview of all the different sensors we have on board. Our most recent flight, I mentioned we've flown 3 times. Our most recent mission just landed, and it actually did 2 laps around Antarctica for 32 days. So we're really excited to report some of the dosimetry and biology in the coming days. Of course, everything's a little bit slowed down right now, as most of you can certainly appreciate with COVID-19, but we'll eventually get that data out. In closing, I just want to point to a couple of other articles that you can take a look at if you're interested in designing balloon payloads. In this astrobiology paper in 2017, again, we characterize how to build this payload. And I also I want to thank Eric Benton at Oklahoma State University, who's really helped me understand the flavor of ionizing radiation, and especially how it compares from an Antarctic balloon mission to, say, the International Space Station. So while we don't have our EMIST results yet from Antarctica, take a look at the light blue line from a tracer Antarctic balloon mission that flew several years ago. And you can see that the dosimetry is really interesting compared to the ISS. And again, I'm trying to argue that you can actually get space radiation without going into low Earth orbit or beyond. Here's another way of looking at that same data. So in this case, tracer Antarctic balloon results are depicted in red, and I'm pointing at the very bottom again to Hassler et al.'s measurements of what the dose rate looks like on the surface of Mars. And that agreement is pretty good. So in conclusion, I want to just finish by helping you understand how to get on board, at least through the NASA Balloon program and their primary contractor, the Columbia Scientific Balloon Facility. They have an annual application, and unfortunately, the call just came out in April, but if you take a quick look, you may be able to get in before the deadline. Otherwise, every April, expect this announcement to come out. They'll basically ask what you're flying, if you've flown before, and what your requirements are. And that's the easiest way to get on board. You can indicate at that time if you have funding or if you don't, or if you're interested in being a piggyback or a mission of opportunity as well. And that's how we've done all of our science flights to date. You keep your balloon payloads simple, and you got a really good chance of getting on board due to the heavy lift capacity of these systems. So I want to just show one other avenue through NASA. The STMD, or Science Technology Mission Directive, also has a quarterly call for flight opportunities, and that includes balloons. A couple of commercial providers to NASA that fly over the United States as well can give you a lift. And so take a look at that website as well if you're looking at some additional opportunities. And I'd be happy to answer any questions. I want to thank my tremendous colleagues at the Kennedy Space Center who did most of the engineering of the payloads that you saw depicted. And I especially want to thank our DLR collaborators for some of their help and expertise on the dosimetry measurements that we're excited to be reporting in the coming years. So that's it, Jeff.
Wow, thank you so much for that presentation. I think that this is a resource that many individuals are not familiar with. So I'm sure we have some questions. While we get ready for Dr. Weil's presentation, I guess the question that I would have for you is, in terms of the payload capacity, it's limited to microbes at this time, maybe microbes and plants. Is that true? And is there any plans in the future for doing, say, something like rodents?
Right. So particularly for the Antarctic missions, because the handover times are so long, like we send our payload down in July and don't usually launch until December. Payload returns at earliest in April. It does help to have a passive, dormant experiment like microbes or plant seeds. That said, we're making strides. The Mars box payload that I showed earlier in the presentation is a pressurized system and the central component where the biology resides, so you can fill it with gas. You could presumably have liquid cultures. And so we're making strides, but scaling up to the idea of hosting rodents would require some more substantial investment from the scientific community and our programs to be able to accommodate animal habitats. I don't think it's an impossible idea, but it will take some time to get there. I will say some of the commercial providers, Jeff, that I mentioned through the Flight Opportunities Program, both of those providers in the United States, I won't name them, I'm not trying to endorse anybody here, but their idea is to actually carry tourists in pressurized capsules to be able to safely view the edge of space while sipping champagne, right? So in theory, some of those capsules, once they're human rated, could also carry some animal experiments. So that's also a possibility too.
Well, thank you very much for that. We're going to be coming back to some of the questions that people have posed regarding the capacity to use tissues on a chip, as well as different types of bacteria, and sort of the radiation profile again. So again, thank you so much. We'll come back. And so for our last presentation today, before we open it up for much more questions, we have Dr. Weil.
Okay, Jeff, can you hear me?
Yes, I can.
Okay, excellent. Well, thank you for the invitation to describe the neutron facility at CSU. And I would like to put in a plug here for Guy and for RareF. I've done experiments with them, and they are phenomenal to work with. And I also appreciate the last presentation. I'm trying to picture my mice sipping champagne on a balloon. That'd be interesting. So, talk about some chronic neutron radiation exposures. And I'm going to tell you about using low dose rate neutrons to simulate space radiation exposure, tell you about the facility at CSU, and if I have enough time, I'll go ahead and tell you about some of our early findings. I don't have to describe space radiation for this audience. I'll skip through this and go to read. So space radiation exposures are going to cause adverse health effects. The one I'm going to focus on here is cancer. And so cancer risk is increased. And how much of that would be acceptable? What level of fatal cancer risk? NASA actually has an acronym for it, READ, risk of exposure-induced death. And the READ for fatal cancer is 3% at the upper 95% confidence interval. So how do you calculate exactly how much of an exposure an astronaut's getting? Well, that's done with a model. The model is called NSCR 2012. It's continuously updated, and it's basically an epidemiologically based model where you're extrapolating data primarily from the atomic bomb survivors to the types of exposures that astronauts would receive. So there are uncertainties involved in the model, and if you want to get an idea of what the risk would be, or the calculated risk for, say, a Mars mission, here is some work from Frank Cucinato and Janice Huff, and you can see that the point estimate for, let's see, this is a female never smoker, is in excess of 3%. And the 95% upper confidence interval is well above 3%. So what you'd really like to be able to do is to cut down on these confidence intervals. And the major things that contribute to the uncertainty that lead to those confidence intervals are radiation quality effects. So going from low LET exposures, such as what the atomic bomb survivors were subjected to high LET exposures, and also dose rate effects. So the atomic bomb survivors were exposed to an acute exposure, instantaneous, whereas astronauts are exposed to chronic exposures. So in terms of the doses and dose rates, David showed some of this data, and the critical ones here are the dose rate for galactic cosmic radiation in transit in deep space, say to Mars, is going to be about 0.5 milligram per day. Once you're on the Martian surface, you got a bit of a carbon dioxide atmosphere. You got the planet blocking out, providing shielding for half the field. So you're down to about half that, down to about 0.2 milligram per day. Now, why are dose rates important? Well, this is data from low LET exposures. And what you're looking at here is the incidence of myeloid leukemia in mice that have been irradiated with different doses of radiation. And you can see that you get a rapid increase in the incidence of myeloid leukemia with dose. and it plateaus or starts going down at about 3 gray. But those are acute exposures. If you do a chronic exposure instead to the same total doses, you get what's called dose sparing, so you get fewer leukemias. And the thought has always been that you're going to get this type of dose sparing. Now, that's a low LET exposure. This happens to be to X-rays and gamma rays. Does that hold true with high LET exposures? And the answer is that there's some epidemiological data that it may not hold true in all cases. And those data come from humans exposed to alpha particles, which have high LET effects, medical exposures and occupational exposures, and also some experimental work done in rodent model systems in which mice are exposed to fission spectrum neutrons, which is what fission spectrum neutrons. So here's an example. This is an exposure to californium-252 neutrons. This is work done by Bob Ulrich, and you're looking at murine mammary tumors. And if you look at an acute exposure, this is the incidence of the mammary tumors with dose. But if you give it as a chronic exposure, you actually get more tumors. That's referred to as an inverse dose rate effect. And like I say, you see it in some epidemiological examples in humans, and you see it in some experimental models in mice, but you don't see it all the time. Also, I'd like you to note the dose rate in this particular experiment. It's 10 milligray per day, or about 20-fold higher than what you'd see in deep space. So we decided that it would be good to have data at space-relevant dose rates, and so we were going to use fission spectrum neutrons for this. And the rationale is that data are needed from animal radiation carcinogenesis studies of high LET exposures delivered at low dose rates for periods up to or exceeding a year. And I guess I'll go into this. The relevance of using fission spectrum neutrons for this type of work. First of all, neutrons are not found among space radiation. Neutrons have a lifespan of about 15 minutes, and the universe is older than that. But neutrons are generated when heavy ions strike a spacecraft hull or the occupants of the spacecraft. So there are neutron exposures to astronauts. And if you look at the integral neutron spectra, for which you would see inside of a spacecraft, this would be inside of a spacecraft with 2 grams per centimeter squared of aluminum shielding. This is 20 grams of aluminum shielding. And this is what you would see from exposures to fission spectrum neutrons, like the mouse experiment I just showed you. So you can see the lower energies are covered pretty well, not the higher energies. You can also use neutrons as a model for the high LET effects of HCE ions. So neutrons, when they strike protons or atomic nuclei, say within the bodies of a mouse or a human, they generate recoiled protons. And if you look at the LETs of these recoiled protons, they sit about right here at about 70 keV per micron. And for those of you who do radiation experiments, I have some other exposures that you might be more familiar with here. So this is 1 GeV iron, the go-to, or used to be the go-to ion at the NASA Space Radiation Laboratory, radon alphas. And this is the dose average LET within a spacecraft in deep space behind different levels of aluminum shielding. So we decided that we were going to build a low dose rate neutron irradiator. We did this out on the CSU, Colorado State University Foothills campus. This is a picture of the campus. And sitting on the campus, we have a few of these buildings right here. We call them blockhouses. And they were built during the 1960s to study radiation effects in irradiated animals. And they were decommissioned in the early 1970s, and they've been used as storage buildings out there. So we got a hold of one of these blockhouses. They've got 30-inch thick poured concrete walls, so they're pretty well shielded. We went ahead and did renovations on the interior, put in good HVAC systems, and got them certified for long-term animal care. So what you can see in this photograph of the irradiator room within a blockhouse is the irradiator itself. It's the blue thing right here. Some of you will be familiar with this design. It's a panoramic irradiator. You have a shielded area down at the bottom, and inside of that shielded area is the source. In our case, a chunk of californium-252, 80 micrograms of it, and it sits right here. From outside of the irradiator room, you can turn on a switch which will drive an air piston, which will raise that californium-252 source up into this tower right here and allow it to irradiate the surrounding room. In front of where the source sits, we have an attenuator. It's the part in yellow right here. And it's a lead attenuator which cuts down on gamma rays, which are also emitted from californium-252. So most of the exposure comes from neutrons. And of course, surrounding that irradiator, we have a circle of cage racks with cages. And when this building is fully occupied, it contains 900 mice and up to 60 rats. So looking at what those animals are exposed to when the source is exposed, most of the dose, 80% of it, comes from neutrons with lineal energies in the neighborhood of 100 kV. And the rest of the dose comes from photons. I believe that Jeff also showed this image here, and it shows LETs by fluence in space inside of a spacecraft that's generated by a GCR simulator beam at the NASA Space Radiation Lab, which is really the best place to do work on radiation quality. And also what you see from fission spectrum neutrons, and you can see this is very similar to what Gus is seeing as well with his accelerator-generated neutrons. So if you're interested in the nuts and bolts of the facility, there's an article online in IJRB that you can go to, and I can send you a copy of it if you'd like. We've had a number of projects that have been completed or are still ongoing at the facility. These are carcinogenesis studies and also studies on cardiovascular effects and on central nervous system effects. And you can see we've had a number of different institutions take advantage of the facility: Georgetown, UC Denver, Loma Linda, UC Irvine, Eastern Virginia, Eastern Virginia Medical School. In terms of services, we've helped outside investigators get approved CSU animal care and use protocols. Some people like to actually work on site, particularly during ski season. And for them, we can get access to the CSU network. If they need tech time, they have access to veterinary residents. CSU is a veterinary school, and also to CSU hourlies. So you can have people around who can help with tissue collection and processing. I should also point out that adjacent to about 100 yards away from the blockhouse, there's a facility called the Regional Innovation Center, So we have good equipment there and good procedure rooms, which investigators can also access. In terms of the availability, we generate neutrons with a radioactive source, which has a half-life, which means that if we expose animals to a milligray per day, when we start off with a fresh source, that source will give you a milligray per day in 16 hours. So essentially, the animals are exposed for 16 hours, and then the source is retracted, and you now have 8 hours to do husbandry. But as time goes on, you have to leave the source exposed for longer and longer periods of time. So at the end of 400 days, what you end up with is animals being exposed for 21 hours and only 3 hours to do husbandry. Given the number of animals that are kept in the facility, that's the time to go ahead and shut everything down and replace the source. And we're in the process of doing that right now. We've obviously been held up by the current COVID pandemic. We had hoped to have it back up and running by September. That time will be pushed back a bit now. I'll show you 1 or 2 bits of data. These are some data from biopsy female mice exposed to these low dose rate neutrons. People have been skeptical about whether you'd see any effect at all. These animals were followed until 800 days of age for tumors. That's why you see the Kaplan-Meier's all ending at about 800 days. And we don't have histopathology on them yet, but most of the deaths were due to tumors. And what you can see here is these are survivals for mice that are unirradiated. If you go up to 400 milligray, which is not a particularly high dose, you end up with about a 10% decrease in median lifespan. There are also results from cardiovascular effects, and this is work done by Mariam Burma at University of Arkansas and Adam Chacot here at Colorado State. And you can see that you have a thickening of the left ventricular walls in these mice. And that is reflected in diminished left ventricular systolic function. There have been neurocognitive and neurodegenerative experiments run out in the facility as well. Those are now published, work done by Rich Britton and Charlie Lamoli. And we've even had a group from University of Colorado in Denver, maternal fetal medicine, look at pregnancy outcomes in mice that are exposed to low dose rate neutrons while they're pregnant. And so the work that I just showed you, the construction of the facility, our physicist is Tom Borak. We've had a couple of graduate and undergraduate students work with us. And we've also had help reviewing the dosimetry from Lawrence Halbern at University of Tennessee and from Adam Russick at the NASA Space Radiation Laboratory. And of course, we've had funding from NASA and some internal funding from CSU. So with that, thank you, and I'll be happy to take questions.
Thank you very much for that presentation. Sort of one of the things I kind of want to tie together, yesterday's discussion, looking at even neutrons clinically with some of the work that you've done in collaboration with other investigators at your facility in terms of CNS effects. If I remember correctly, Dr. Sengel referred to neutrons as basically another beast altogether in terms of comparison with charged particles and photons in terms of CNS effects. And so I think for the audience, because these are recorded, it would be good if you wanted to at least check out the publications that have come out of this facility looking at CNS effects and comparing that with some of what's been known, at least in terms of neutron exposures in the brain. And if you would, before we get into the questions, could you summarize any of those findings in terms of CNS effects with these neutrons?
I would not dare do it. You know, I'm a cancer guy, and I don't do brains.
Well, those publications are available for those of you who can look this up. Again, these are all recorded. So thank you very much for that presentation. What I'd like to do now is address some of the questions that we've had. And if you have any other questions for any of the panelists, please feel free to ask. And we're going to start by going back to Dave Smith's presentation. There were a number of questions about, again, the capabilities of the platform and the payloading. And is it possible to do tissues on a chip, or how would it work for BSL-2 bacteria? And if he could comment on some of that.
Yeah, okay. Regarding the second part, BSL-2 or any biologicals do have to go, if you're flying with NASA, have to go through NASA safety offices. So Bloom Program Office is managed out of Weil Flight Facility, and their safety team will take a look at SDS sheets, so on and so forth, and the containment measures that you have, quite like it would be if you were flying an experiment to the space station. So in short, yes, it can be done. Does it require a little bit more paperwork and processing for anything above BSL-1? Indeed. With Antarctica, there's also another layer of complexity with environmental permits. You have to give both NASA and NSF. NSF is actually the agency responsible for running operations down south in collaboration with NASA with the balloon launches. But the NSF has to clear all the environmental paperwork, not only for Antarctica to ensure you're not leaving anything behind in the event of a mishap that anything gets dispersed, but a lot of these payloads go through New Zealand, so you also have to do environmental paperwork through New Zealand. All of that can be done. Regarding tissues on a chip or any other types of experimental designs, I think it really just depends on how you build the payload architecture. I can count on one hand how many biological payloads have flown on large scientific balloons, and so we're really in chapter 1 of what I hope will be a very long book of experiments to fly. And compared to the cost of building and proving out a payload to fly to the space station or deep space, it's pretty accessible and affordable and fast. And so if you're thinking about doing tissues on a chip, I would encourage you to look at payloads that have flown to other places, not on balloons, but to space station or free flyers, because those are pretty amenable to balloon operations. Like a lot of you are probably familiar with the CubeSat configuration. You know, that would be an easy way of doing your science and just attaching it to a balloon gondola without having to do much to get it ready to go.
Great. So it does look like there's a lot of capabilities for different on the payloading systems. There's a number of opportunities to study various model systems.
Yeah, very much so. I think the field will grow rapidly and be able to accommodate more types of biology in the coming years. And if we, the collective we, right, NASA's programs and stakeholders were interested in flying a dedicated biology mission, you could have up to 6,000 pounds worth of biology and hardware on a single mission. We're not there yet, but it is within the realm of possibility. It depends on program priorities.
Now, Dr. Garty, in terms of your facility with the microbeams, now, are those reserved primarily for single cell studies or tissue level studies, or are you also performing them in rodent studies? Is that available to investigators?
Well, the issue with rodent studies is at the moment our microbeams, except for the protons, don't really have enough penetration to go anywhere inside an animal. So we're in the process of setting up a facility that would allow doing these sort of experiments primarily using transdermal windows. Because even with the best beam we could ever get, we probably wouldn't be able to penetrate more than 1 millimeter into tissue. So with the energies we have available, it's primarily looking at the thin targets.
And another question regarding your facility was, has there been a systematic study of the cellular ion channels for conformational changes that would impair function with carbon radiation? Has any studies looked at how carbon radiation can alter those ion channels and how that can relate to impaired function?
I do not know. We haven't done any carbon irradiations yet at our facility, and I don't think I've heard of anything about that, but I'm a physicist. I don't follow the ion channel literature too closely.
Okay. Here's another question. Do you, for my features, to Dr. Smith, do you have any insights on how to control for circadian effects of continuous sunlight on long-duration balloon experiments?
Yeah, I mean, so a couple of considerations, right? Continuous sunlight was a feature of polar balloon flights. So the Antarctic summer allows for continuous sunlight, but even in that scenario, you could design your payload to, say, have a shutter like our system where you could, you know, simulate the amount of exposures and some diurnal effects. Domestically flown balloon missions or missions flown from other parts of the world can linger both in the day and night. The balloon tends to lose a little bit of altitude during the night. But you, on a mission, say, from Sweden, launch from Sweden or launch from New Zealand, would be able to experience natural diurnal swings. And so it just depends on where you fly from. And if you're on an ultra-long duration mission from the Antarctica, you could control the light exposure based on your payload design.
Great. And for Dr. Weil, here's a question. So, you know, the combination of spaceflight hazards has been shown to be of particular interest and may pose a greater risk than those individual challenges alone. So that would be combined, say, simulated microgravity via tail suspension with radiation. Is your facility set up so that you can do those kind of combined studies?
Yes and no. So we've done combined studies. We also have, I didn't show you this, we also have a facility where we do low dose rate gamma rays. And we've published studies where we do hind limb suspension and low dose rate gamma ray exposures for protracted periods. It's a very simple matter to set something like that up within the neutron facility. However, my understanding is that you'd have to redo a lot of the dosimetry in order to do that, and that can be fairly time-consuming.
Yeah, just as a note, I'd just like to say my very first radiation study ever was in collaboration with you all through my Ph.D. advisor, going out doing some of those low-dose gamma ray studies.
Excellent.
So, yeah. Another question for Dr. Smith. For those of us who have done studies on ISS or on shuttle, know that there's always this concern about payload turnover times, right? Can we be present for the turnover? Can you comment on the turnover times, considering you can do these really long experiments in Antarctica and relatively shorter, but still prolonged studies out of New Mexico?
Yeah, good question. So let's talk about the domestic flights from New Mexico and Texas. Usually what happens is there's an integration of the payloads, and then after that occurs on site, you launch as soon as the next morning, depending on the wind conditions. Now, the wind conditions are really key to getting anything off the ground, and so if you're lucky, you could be on site with your biology and the next day be flying into near space. And depending on where the system comes down, you may have the payload back in your lab within 24 hours. And that's the best case scenario. However, with the winds being what the winds are, it's usually a 3 to 4 week, I would say on average, time from when your payload in biology is ready to go to when you actually fly. So that's something that you have to consider, either figuring out a dormant model system to work with or something that's going to be stable or replaceable over that 3 to 4 week period for a domestic flight. And I talked about the handover times with Antarctica. That's just substantially more difficult. The other thing to know is you're considering a piggyback payload to go to Antarctica is you don't travel with it. As you can probably imagine, NSF and NASA only have a limited number of seats, and that for scientists to actually go down and spend the season in Antarctica. So those seats tend to be reserved for the primary payload. In the case that I showed you, we were flying on board the Super Tiger mission. Super Tiger scientists were the primary payload, and we've got to travel with the system. Piggyback payload like ours, we don't. We hand it over, and of course, we can fly or not fly. We can use various ground controls, both that stay in the lab and travel with the payload that don't fly, so that we can kind of disentangle the effects of these long handover and return times. So in short, it's a little complicated with it, but so are any experiments you launch at the space station. So it can be managed, but you do need to think carefully about the types of biology that you're loading in the system and what happens with delays.
All right. Well, I would like to thank all of our speakers today. This has been a fantastic forum. Many of the participants have reached out in various ways and asked for contact information from the particular speakers. And I would advise you can take a look on TRISH's website, in which all of these videos will be posted, and you can get the contact information from the speakers off of the recordings, and the information about the speakers should be available there as well. So I think we're going to wrap that up for today. I'd like to thank our speakers, Dr. David Smith, Dr. Guy Garty, and Dr. Mike Weil. It's been really informative, it's been fun, and thank everyone for attending today, and hopefully we see you tomorrow. So see you tomorrow at